Hi, Dr. Stansel. It's Ashley Allen. Today is September 24th, 2018. Yes, I'm starting week 5 early. There's a good reason for that. So last lab, we spoke about microbial testing for identification purposes and classification purposes. So same thing, we're doing some biochemical testing, although this one's different. This is carbohydrate testing. And so in this lab, I have the 6 phenol red broths. They've come out of the refrigerator. I've begun breaking up the 3 different types of carbohydrates we have, which is fructose. They come from carbs and fruits. Glucose, which is from plants, and I read the metabolism of animals. And then there's mannitol, which is a new one for me, mannitol, which is a crystalline alcohol produced by plants. So we have these little tiny vials that we had to split up between each one, right? So there's 2 for glucose and 2 for fructose and then 2 for the mannitol. So we have 1 more mannitol to do, and I will do that with you. But before I do that, let's talk about the purpose of carbohydrate testing. Basically, carbohydrate testing determines the byproduct of fermentation, and so it differentiates among the bacterial species. So again, fermentation is carbon, hydrogen, and oxygen, right? When things happen without oxygen, it's fermentation occurs, so it's anaerobically. So now we're just another way of testing microbes. And again, this is important for classification and also to see what kind of environment our microbials live in, or how they get by and thrive. So I'm gonna face the camera down and put on my mask and everything. So let's bring this down here. Okay, you see my 6 tubes. So I'm gonna set this here, put my mask on. So remember, we heat up the edges of these tubes just to prevent contamination. We want that to happen. As I sneeze, excuse me. And then we have just a little mannitol left, and we're going to carefully place it in this last vial. Kind of had to shake them up. I noticed, probably because they're carbs, that they stick to the bottom. There we go. There we go. So we're going to place that in here. I'm going to just do a slight mixing. It didn't say to do this, but it didn't say not to do this. So just so the pieces are not in whole. Then next, I have my Durham tubes, which are pretty impressive in my mind. And we're going to place them inside of our larger phenol red broth, because remember, fermentation creates gas. So we need to put these in here, open side down. Now, I just know already there's going to be air bubbles in there. And sure enough, look at that. That means there's air in there. So I'm going to close this, and to be honest, I had to practice this. I used a practice tube because I wanted to make sure I knew how to do it, and there's still a little bubble in there. Can you see that? So I'll just kind of play with this gently and get that bubble out, but I have to do this to all 6 of them. So after we're done doing that to all 6 of them, I have my broths set aside here, and for each one of these, as you know, there's glucose, but here's glucose, and one is S. cerevisiae, and the other one is S. epidermidis, right? So each one of these are going to have their own broth in there. We're going to do about 4 drops of broth there, and then we're going to let them sit there and incubate for 12 hours, and then I'll get back to you with the results.